Hi guys, this is David Villar and welcome to the second video on toxicants that affect the liver. Here we will discuss blue-green algae and plants containing pyrrolistidine alkaloids. A few general concepts to blue-green algae is that there are up to 2,000 species and most of them are non-toxic. There are 95 known identified toxins and the number of cells do not correlate with the concentration of their toxins, and they need to be lysed to be released to release their toxins in an environment like the stomach contents or the rumen. Toxic blooms tend to occur during sunny days in stagnant bodies of water with high nutrient content, especially phosphate. And commonly animals are exposed to potentially toxic amounts or when pastures or holding pens uphill have fertilized the pond with animal waste, and the wind has concentrated the bloom near the side of the pond where the animals drink. This is what we see on this image. The algae have buoyancy, which leads them to accumulate at the surface of the water, and then the wind will drag or transport them horizontally to the side of the pond where they can build up in thick scums. There are numerous toxins, but we will discuss the best known of all of them, which is microcystin. Microcystis fresh blooms are green, and microscopically the cells are arranged in globs containing hundreds or thousands of marble-like cells that are grouped in a common matrix. The individual cells are approximately the size of a red cell, and the toxic principles are a group of similar cyclic peptides with about 7 amino acids, and there are up to 100 different varieties of these cyclic peptides that have been identified. Now the mechanism of action is shown here with the liver cells being the primary target because they are unique in that they possess a specific transport carriers in the membranes, and that allows for the penetration or the uptake of the toxin. They inhibit 2 phosphatases resulting in excessive phosphorylation of a specific proteins, including the cytokeratins, which are the major proteins that make up the cytoskeleton. What happens next is that the hepatocytes round up due to the loss of microtubules and actin microfilaments, and this causes the sinusoids to disrupt, and the red cells fill in the dead spaces in the liver, which, as you know, which as you can imagine will be very hemorrhagic. The animal dies with acute liver failure and severe shock. We mentioned the microcystins, but there are actually other toxins that are produced by different types of blue-green algae. For microcystin, the syndrome may last less than 24 hours from the onset until death. The animals tend to develop an acutely lethal syndrome that starts with GI signs and are followed by weakness, pallor of the mucous membranes, unresponsiveness, and death, obviously. The 2 other toxins listed here are called anatoxins because they are produced by Anabina species, not microcystin ones. They are not hepatotoxins, but they are neurotoxins. The first one, anatoxin A, is a nicotinic depolarizing alkaloid and produces a syndrome similar to succinylcholine overdose. There is a rapid onset of muscle rigidity and tremors, then paralysis, cyanosis, and death from respiratory paralysis. With anatoxin AS, this is actually the only naturally occurring organophosphorus cholinesterase inhibitor. The word S stands for salivation. So just as we saw with cholinesterase inhibitors, there is going to be buildup of acetylcholine, and that is going to cause excessive salivation, GI hypermotility, diarrhea, urination. And the nicotinic signs would be those of tremors, incoordination, paralysis, and terminal respiratory paralysis. On this slide, we have an example of an acute microcystin toxicosis in a dog. The history was that he spent the day with the family in a lake in Kansas. The same night, he developed a greenish vomit and diarrhea, and the next day he was taken to the university hospital and was already recumbent and responsive with abdominal pain, melena, and an icteric plasma. It was treated with dextrose, fluids, vitamin K, plasma, and acetylcysteine antiemetics. And in spite of all the efforts, he developed anuria and was euthanized at 36 hours post-exposure. I like this case to show what a CBC and chemistry panel will show with an acute liver failure. The packed cell volume was 70%, the clotting times were markedly elevated, glucose was lower than normal, and the liver enzymes were all through the roof, as well as bilirubin. Now, when they necropsied the dog, the liver was swollen and contained huge amounts of blood. On histopath, the picture was of a complete loss of hepatocytes of the entire liver lobule with replacement by red cells. In the vomitus of the dog, they identified the round colonies of microcystin aeruginosa, forming circular mats of closely packed cells. They also confirmed the presence of microcystin with sophisticated lab analysis in the liver and the stomach contents. So next we will discuss plants containing pyrolicidin alkaloids. There are three major families. Some of them may have thousands of species, like for example the Synecio genus, and obviously not all of them are toxic. In general, under normal conditions, they are avoided by grazing animals, but they may be eaten if there is no better forage, for example during drought conditions. And it's typically a chronic toxicosis that results in progressive liver failure. Now, there are numerous cases reported throughout the literature of poisonings in horses and cattle, and so it's going. Probably the most commonly reported are the Synecio ones that are a genus of flowering plants in the daisy family that include ragwort and groundsels. It is actually the largest genus of all the flowering plants, and the flower heads are normally rayed with the heads borne in branch clusters and usually completely yellow, but they can be also of other colors. We see here a field that is completely overtaken by the South African ragwort, Synecio inaquidens, which has now actually colonized many other countries worldwide and is considered an invasive species in many grazing fields. Here we have another plant also containing pyrolicidin alkaloids. This is actually in the pea family, and it's called rattle pods because the seeds become loose in the pod as they mature, and they rattle when the pod is shaking, just like a rattlesnake would do. And the genus includes over 700 species, and some of them are cultivated as crops to be consumed by humans throughout the world. Now, you may wonder, what are the mechanisms of action? Well, there are about 100 known pyrolicidin alkaloids, and only a few have proven to be toxic. They are rapidly absorbed from the intestines and thereby expose the liver first via the portal circulation. Once in the liver, there is conversion into a pyrrole derivative by microsomal enzymes, which presumably alkylates macromolecules. It primarily cross-links DNA strands, and that's going to impair the cell division. So what happens is that the hepatocyte cannot undergo binary division and keeps growing and growing, unable to divide. This is somewhat unique when you look at the liver microscopically. You see megalocytosis, or huge hepatocytes, and you may wonder, well, what the heck, the bigger the better. But those hepatocytes will keep expanding, sometimes up to 10 times their usual size, and when they reach a critical mass where things cannot get where they should, then the cell dies. And with the death of a large number of hepatocytes, the necrosis triggers a regenerative response, which is accompanied by bile duct proliferation. So the three primary lesions of pyrolicidin alkaloid poisonings are going to be hepatocytomegaly, fibrosis, and bile duct proliferation. And these lesions are both dose and time dependent. On this image, both the fibrosis and the biliary hyperplasia are very clear to identify, to identify. It will eventually reach a point in which most of the liver reserves are depleted, at which time the liver function collapses and the animal suffers from a generalized insufficiency syndrome of the liver. So what are the clinical signs expected? Well, with acute intoxications would be very rare events because the plants have very poor palatability, and that would make a rapid ingestion of large quantities very uncommon. So what we're going to see most of the time is chronic exposures, and just as we saw, the liver reflects the cumulative and progressive effects of repeated ingestions. So the signs may take up to weeks or even months to appear after the initial exposure. In horses and cattle, the clinical signs include loss of condition, anorexia, dullness, and constipation or diarrhea. Tenesmus and passing of blood-stained feces may be followed by a rectal prolapse, especially in cattle. And horses will be more frequently showing nervous signs because of the hepatic encephalopathy. For example, head pressing, yawning, aimless wandering. That has also been called a sleepy staggers or walking disease. And you may see ascites and icterus sometimes, but not always. So at this point, there is so much liver damage that treatment is obviously unsuccessful, and they will die within a week of the onset of signs. Now let's describe a case of Tansy ragwort poisoning in 5 horses. This incident took place in Belgium. In these cases, they were believed that the animal had ingested the immature seedlings of the plant that are shown with an arrow on the picture. They all develop some or all of the clinical signs that we described previously, and were taken to the vet school in Belgium, where they were given symptomatic treatment, including fluids, blood spectrum antibiotics, flunixin, meglobin, and easily digestible diet. In spite of all the efforts, the conditions of the animals deteriorated and they were euthanized. So they did a range of laboratory tests that are kind of useful to diagnose pyrolicidin alkaloids. The most relevant was probably ammonia, that was very elevated, and as you know, ammonia is produced in the cecum and the column by bacterial degradation of proteins and amino acids. It then diffuses into the portal circulation and is transported to the liver, where it should be converted to urea. Hepatic encephalopathy actually reflects a failure of this important detoxification process and is caused by extensive liver cell necrosis. Now, the low levels of serum urea reflected the reduced hepatic synthesis from ammonia. Total and unconjugated bilirubin levels were also good markers of liver damage because there is reduced hepatic uptake and conjugation of bilirubin. They also did some liver enzymes like gamma GT and alkaline phosphatase. And also the elevation of bile acids is another indication of reduced functional mass of the liver. So this is the last slide for this presentation. On the next one, we will continue talking about more toxicants that affect the liver. So thank you for listening, and until next one, take care and bye-bye for now.